Hello, everyone. I will start by thanking Katerina and NBIC for inviting me to this webinar. And as Katerina said, I am head of R&D at Labskin. Labskin is a company currently based in York that provide tissue models, skin tissue models, full thickness, contract research for skincare, cosmetics, and some pharmaceutical companies. So I will talk about a little bit today about what is Labskin, what is different between about Labskin and other tissue models, how we have worked with vaginal microflora, and also I was invited, Katerina was asking me to talk about our new development with something called Skin Trust Club, which is related to using the skin microbiome as a tracking device to look at skin health. So I will focus more at the end on the Skin Trust Club, but I will talk a little bit about some models we have developed for some of our clients working with the skin microflora. The first thing to say is Labskin is a skin model, a full thickness skin model, so it's not a mucosa. So it contains a dermal layer and epidermal layers, contain fibroblasts and keratinocytes. The fibroblasts are embedded in a fibrin matrix, so there is no exogenous collagen, and that fibroblasts produce their own collagen and their own extracellular matrix, and that allows us to see the keratinocytes on top and then fully differentiate, where you can see basal membrane, basal layer, stratum spinosum, stratum corneum, and stratum granulosum. That gives us a very, very dry surface with a very good barrier function that allows us to colonize with microbes. So one of our unique selling point is Labskin. We can colonize Labskin with the skin microflora and even with whole microbiomes, as I showed you later. This has been developed during the last 15 years, and we have worked with lots of the biggest skincare manufacturers in the world. And also, the skin model has been validated, independently validated for some of our collaborators and some other research studies, working on different aspects, for instance, in transepidermal permeation of drugs using MALDI imaging, or in infections, and using mass spectrometry to identify bacteria on those infections or burns and some other aspects. As I say, we are a contract research organization. We set ourselves in the preclinical space, in the preclinical test, but we can also do some clinical trials. One of the things we do is clinical trials on the microbiome. We have something called remote clinical trial, which we can send a swab to participants. Then we collect the microbiome from those participants, and that swab is sent back to our facilities, and we do something called hybrid trial. So we can test skin products in both the volunteer, but also on the microbiome of the volunteer that can be replicated several times using our skin model. So we try to reduce the amount of variability of studying the skin microbiome, but cloning or replicating the same microbiome several times in different pieces of Labskin. Reducing variability. We are animal-free. Our skin model is stable much longer than an ex vivo model. They have some, for instance, we can keep Labskin growing for up to 30 days of air liquid interface. We normally use it when we study skin. When we do skin studies at day 12 of air liquid interface. But also we can use it at shorter times of air liquid interface to mimic some sort of sensitive model, and that's what we do for when we study no vagina, but vulva or external labia microflora. And these are some of the endpoints that we can produce for our clients. We can do classic microbiology, colony counting. We can look at immunology using ELISAs and gene expression. We can do transcriptomics, metagenomics for microbiome. We have sequencers, and we have multiplex ELISA. We have fluorescent microscopy, and we even have mass spectrometry. So there is a huge amount of endpoints that we can produce. So about the vaginal microbiota, as I say, We don't have a mucosal model. We work with a sensitive model to study the microflora on the external labia or the vulva. I'm not gonna enter on description of the vaginal microflora since there are two speakers already that have talked about this, but just highlight that most of the infections happening in the vagina are produced by yeast or bacteria that are already there, and they are commensals, and that they are opportunistic pathogens with conditions, physiological or ecological condition change, they become pathogens, apart from STDs. And that's something that in the microbiome space called as named as dysbiosis. So things that we can do on our skin, sensitive skin. For instance, we can study vulvovaginal candidiasis, so the infection by Candida albicans, one of the most common infections in the vagina and the vulva. And we can do that by simply putting Candida albicans in different conditions of observed infection. We can do colony counting, and then we can look at the expression of several cytokines that are indicated of disease. So IL-1 alpha indicate damage. This is known to be triggered by irritation, but also mechanical damage, while IL-23 is more specific of infection. It responds quite well to aureus, but we have data showing the response also to the presence of Candida albicans. We can do the same with a bit of complication on the biofilm as a biofilm model. Of course, Candida albicans and Staphylococcus aureus are probably not the most common polymicrobial, or this is an interkingdom biofilm, yeast plus bacteria. As I say, it's probably not the most common thing to find in the vagina, but it was for us to study as a model to look at the efficacy of drugs against biofilm formation on this type of tissue. Again, we can see the infection by using immunohistology, also colony counting. And additionally, we can see the response from the tissue, from the host, by the expression of cytokines. In this case, it's a little more of IL-1 alpha because the biofilm was created by making little incisions in the tissue and colonizing with both microbes together. So as you can see, the IL-1 alpha decreased with time, while the IL-23, which is specific to the response of the infection, increased with time, which makes sense for this model. We have also worked with clients to look at these biotic models on the vaginal microflora. In this case, we were looking at a probiotic or a possible probiotics on the skin, Lactobacillus plantarum, and also a prebiotic, which is this product X here. And we did a colonization with these 4 microbes here, Lactobacillus plantarum, Epidermidis, aureus, and E. coli. And then after colonization, we treat with this product and wait for several time points to collect the microbes and also look at cytokine production. As you can see, the product X clearly increased the number of Lactobacillus plantarum with time and decreased the number of the bacteria. And that can be correlated with the production of IL-23. As I said before, is indicative of infection, mainly by aureus. However, this product clearly increased the production of IL-1 alpha, indicating that might be irritating the skin. So it's a bit of a mixed result. We also can test for our clients, and we do this very often, is the effect of tampons in the microbiota. So basically, we colonize the skin with different species, and we test different tampons. In this case, we have a regular tampon, a tampon embedded or soaked into CBD, cannabidiol oil, which is known to have antimicrobial effects. And the consortia here was aureus, Lactobacillus acidophilus, and Candida albicans. But because of the presence of acidophilus, we couldn't count Candida albicans. That doesn't mean they were not there. You can see them in the images that those are that arrows indicate yeast, so there was a small accumulation of yeast, but they were not growing in a great amount. Something that also we do for a lot of our clients is something called preclinical microbiology. So basically, the FDA is going to produce tampons or pads or something that are related to female sanitizers. you need to test for this preclinical microbiology, mainly to avoid that your product increase the production of the toxic shock syndrome toxin by Staphylococcus aureus, and that is obligatory to produce this test. So the FDA doesn't give doesn't provide any protocol, just send you to different papers. So the most common thing to do is a pure in vitro test, where you can put the different several bacteria on consortia. Here we have aureus, lactobacillus, acidophilus E. coli, and Candida albicans all growing together in the presence of absence of tampon in a synthetic medium containing blood, and we can follow the growth of the bacteria and the yeast, how the pH change, the changes in Candida albicans morphology. Candida albicans grows as a yeast and become infected by growing, changing morphological aspect of its growth and become a hypha. So we also look at that by microscopy, and we also look at the production of toxin in the presence of absence of these different tampons. However, this is pure in vitro, so it doesn't take into account the tissue at all. So we were looking to improve this type of test. So we try to use Labskin on this development, and there is some literature showing that the presence of TST1 toxin on in epithelial cells induce the production of two cytokines, MP3A and IL-8. So we try to test that to see if that was possible to use Labskin to do this sort of testing. So in this case, you will see here we have two different plates, one containing Labskin that was treated with different concentration of toxin, and the other one containing different strain of aureus. So the NCTC 13435 is a strain of aureus that is known to produce very little TST1 toxin, while the 11963 is a toxin producer. And then we colonize with those bacteria, and we also colonize tampons embedded medium with those with the toxin producing bacteria in two different tampons. And we analyze the production of these two cytokines, and you can clearly see that it's a clear effect on the production of both MIP3A and ILA in the presence of toxin, but also in the presence of the bacteria that is more toxigenic. And we did a multiplex ELISA to see if we can find other markers related to this. And we can see here 1B, IL-4, 12P70 and TNA alpha. They are clearly induced by the presence of these genes. So we have a model, we think we have a model now that we can use to look at the adverse output pathway instead of the toxin producing itself. So basically what we're looking at here is not the bacteria producing toxin, but how the tampon with the microflora interact with the host. So it's a more complete model. I was rushing about that since other people was talking about the skin microflora and the skin microbiome, because I wanted to talk about the Skin Trust Club. And the title of this presentation is the Labskin Perfect Cycle. Why the perfect cycle? Is because we have the Labskin, this tissue model that can be colonized by microbes, but also we have the remote clinical trials, which is we can isolate the microbes from volunteers and grow those microbiomes, or maintain those microbiomes on our Labskin model. And that is done for clients. So we were thinking, can we do this for consumers? And that's the idea of the Skin Trust Club. So just to give you a highlight on the skin microbiome transplantation, we are able to extract the microbiome, different techniques like screw wash or swabbing. We collect those microbiome. Those microbiome can be frozen. We can sequence them and then colonize with that microbiome onto Labskin, and we can do studies on that. So we have two initial isolates here, and those isolates were replicated several times in two different conditions, and you can see how that microbiome shifts depending on the treatment. And this was done by 7 days, and the initial microbiome, the final microbiome were actually quite stable. We're talking about 16S here. So these are genus with little ability to look at the species, as has been mentioned before. But we know that we can at least recover a microbiome that resembles the initial one, is not significantly different after 7 days. And we do this for clients. So in summary, we can colonize with individual microbes. We colonize with consortia. We can colonize with whole microbiome isolated from volunteers and do a whole range of different experiments and look at different endpoints depending on the preference of the client. So now, what is the skin trust club then? So the skin trust club related to what we were saying here is basically slightly different to remote clinical trial. We send a swab to a participant, a consumer, and then we recover that swab and we do sequencing on that swab. Metagenomics. We use the microbiome to tell things about your skin. So basically, to give you an idea of your skin health using a novel biomarker, which is the microbiome. It's quite simple. As we have an app, you download the app, you order the kit, we send you the swab, we recover the swab, we do the analysis. When you activate the kit, we also take environmental data with the pollution, UV index, and some other type of environmental data. We also have a questionnaire where you can ask a question about, are you taking antibiotics? Do you have a skin condition? Everything is collected as metadata that can be used for the analysis later. You receive your kit, and then you receive in your app, directly on the app, a skin microbiome report with a microbiome balance that we create using well-established bioinformatics and AI models that pick several features that give us information about your skin, and we do recommendation based on that microbiome score. So the technology is based on metagenomics, on the skin microbiome, but also AI modeling. And we collect microbiome data, we collect metadata, age, health, diet, medication, and also we collect environmental data. And all these things can be mixed together to do certain analysis. For now, it is not a medical device, not a diagnostic device, so it's only for skin health, but no a diagnostic. We are working already to use this to do as a companion device, some sort of device that can be used to diagnose severity and establish severity of different skin diseases. And to do that, what we are creating is a huge microbiome database. We already have the biggest skin microbiome database in the world. We have thousands of data points on the skin microbiome. And the skin behind is microbiome analysis. We are already developed a method to look at the metabolites using mass spectrometry, and we are working to do a gene expression from the host part. And I think I'll leave it there. And thank you very much.